The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everyone. It is with great pleasure that I welcome today's speaker, Robin Hyde. Robin was chosen to speak today as part of Rosa's Early Career Investigators Initiative. Robin received his bachelor's and master's degrees in pharmaceutical sciences from ETH in Zurich, Switzerland, and he graduated as valedictorian of his master's class. His master's thesis was the development of a tool for pharmacodynamic modeling of bispecific protein degraders and its application for systems analysis. During his master's work, he interned at Bayer, and he has continued to work there in DMPK preclinical modeling and simulation while he works on his PhD. Robin is working on pharmacodynamic models for new therapeutic modalities, currently focusing on proteolysis targeted chimeras, PROTACs. He applies these models to support drug discovery programs at various stages by guiding medicinal chemistry efforts, planning animal studies, and translating the results to human patients. Robin will be taking questions at the end of the webinar, and you can ask him questions at any time during the webinar by entering the question into the questions pane on your GoToWebinar side panel. And it was my great pleasure to give a very warm welcome to our speaker, Robin Hyde. Robin?
Yeah, thank you very much, Rebecca, for the kind introduction. And let me take the opportunity to tell all of you what an honor it is to be invited here to talk about some of our recent work on the PK/PD modeling of proteolysis targeting chimeras, or PROTACs for short. Just as a short recap about myself, I'm 25 years old, and right now I'm in the second year of my PhD at ETH Zurich in Switzerland. However, as an external PhD student, I'm based off campus with Bayer in Berlin. And therefore, we will be looking at both literature as well as in-house data today, right? And since I'm in the preclinical modeling and simulation group, all of these data and the questions we want to address with them come from the preclinical phase of drug discovery and development. So let's see whether I can change my slides. Oh yeah, I think I do. Yeah. So let's get started. What actually are PROTACs? Well, as I said, the acronym PROTAC stands for proteolysis targeting chimera. So these are drugs that degrade a protein of interest, or POI, that's the red one. However, they can't do that on their own, but they need the help of an enzyme that is widely expressed in human cells, a so-called E3 ligase, to achieve degradation of the protein of interest, POI. And the way this works is that the PROTAC, so that's the drug, binds to both the E3 ligase and the protein of interest at the same time, forming a ternary complex, a complex with 3 constituents. Next, the E3 ligase catalyzes the ubiquitination of the target protein. That is, it attaches a ubiquitin moiety to the POI, and that happens over and over again, right? So we end up with polyubiquitination. Now, at some point, the PROTAC, remember that's the drug, dissociates from the ternary complex, thus releasing the protein of interest, which is now polyubiquitinated. And finally, this polyubiquitin tag on the protein of interest is recognized by the proteasome, which in the end degrades the protein of interest. Right? So PROTACs degrade protein of interest. Great. Now that we know what we are dealing with here, let's get started with the actual modeling. And the first thing we did is we took the cartoon from the previous slide and analyzed it through the lens of the 4 pillars concept to arrive at a more abstract understanding of the mechanism of action. right? So in order for a PROTAC to elicit a pharmacological response, it has to get into the target cells, where it must then bind to the target protein itself, as well as to an E3 ligase to form this ternary complex. In this ternary complex, the E3 ligase then mediates polyubiquitination of the target protein, which is subsequently degraded by the proteasome. And finally, this degradation translates to a downstream pharmacodynamic effect, so we get some sort of biomarker response. Now, today, we will be focusing on pillars 2, 3, and 4. That is the pharmacodynamics, which we will be studying in vitro, while we won't go deeper into pillar 1, so we won't discuss the pharmacokinetics today. So, where does this place us in the grand scheme of things? In this slide, I show the 3 primary applications of preclinical modeling and simulation that we work on here at Bayer. And with our pharmacodynamic modeling, as the topic for today at least, we fall into this first category, where what we do is we translate biochemical or physical chemical data, such as binding affinities, to cellular data, such as protein degradation in vitro. Now, if you're interested in the nitty-gritty of how that works, so if you want to have a look at the underlying equations and the derivations, I proudly refer you to this paper that we published back in January, where we outline all of that in great detail. Here in this presentation, we will instead focus on the underlying ideas and the applied side of things. But of course, you're very welcome to also ask more technical questions in the end, in the questions and answers section. Goodie. So then, the applied side of things. Now, what kind of applications are we talking about? Well, we identified 3 key questions that we frequently want to answer based on in vitro data, and then we designed tailored models to address each of them. The first model allows you to assess a PROTAC with regards to target degradation. If we are not fully content with the performance of that initial compound, we might then want to further optimize it, which is where the second model comes into play. And finally, we also want to know how much degradation we actually need. So we want to be able to translate target degradation to a more relevant downstream pharmacodynamic response, and we can do that with the third one. So, first things first, let's imagine a new PROTAC has been synthesized, and now we ask, how good is it? So, how much degradation is there? Well, the extent of degradation, of course, depends on the concentration, which is shown here on the x-axis in this plot. Therefore, let's start off by quantifying this concentration-response relationship using a mathematical model, which will then afford parameters that we can use to compare different compounds, for example. Currently, the best practice here is to go with the hyperbolic Emax model, which is shown as a solid black line in the main plot in this slide. Now, that well-established model, however, cannot account for the hook effect, which is characteristic of degradation by PROTACs. So it cannot capture what's happening at high drug concentrations, right? Now, what is this hook effect and where does it come from? Let's have a closer look. Now, as you can see, now you can see it. As you can see, this hook effect is caused by the PROTAC, which is on the x-axis in this plot. There's PROTAC concentrations on the x-axis. So hook effect is caused by the PROTAC forming binary complexes. So that's complexes with 2 constituents, rather than the required ternary complexes at high concentrations. So you can think of it like that. If there's a lot of PROTAC around, so if we're on the right of that axis, then every E3 ligase, depicted in blue here, and every target protein, shown in red, they will have their own PROTAC bound to them. So the E3 ligase and the target protein will not have to share a common PROTAC molecule between them, because there's just so much around. And as a result, the E3 ligase and the target protein are no longer brought into contact with each other. Therefore, there's less ubiquitination, which ultimately leads to less degradation. Now, Anyways, to avoid such misfits, we have extended the Emacs model, yielding what we call the hook model. And that's the dashed line going through all the data points. Now, in addition to Emacs in DC50, that's the maximal extent of degradation, Dmax, and the PROTAC concentration of half maximal degradation, respectively, DC50. So in addition to those parameters, which are already part of the Emacs model, this model that we here propose also considers the concentration of maximal degradation, or DC max, and thus allows to fit all the data. Moreover, the new hook model also gives precise and accurate estimates for Dmax. So that's the dotted line at the top in this plot. That's the estimate from the hook model, while the Emacs model tends to overestimate Dmax, which then also leads to skewed compound rankings. And to demonstrate this point, we assessed a bunch of concentration degradation profiles from different compounds. And this plot here shows the results of that analysis, where the same data were always analyzed with the Emacs model and with the hook model. And the estimates for Dmax, so the ones derived from these two models, were then plotted against the highest experimentally observed degradation. So on the y-axis, we have the fitted values, while on the x-axis, we have the real, the observed values. And of course, there's always two model estimates for each observed value, right? Because profiles were each analyzed with both models. Now, as you can see, the estimates from the hook models, so the golden ones, are spot on, while the Emacs model tends to overestimate the Emacs parameter, but it does so in an inconsistent manner. So in some cases more than in others, which messes up compound rankings, as I illustrate in this table here. And that's, of course, an issue when you're trying to build structure-activity relationships, for example, where the Emacs model might give you a false impression of what's actually going on. Okay, what we've been looking at so far was the first major peculiarity of PROTACs, that being the hook effect. However, there's also a second important phenomenon that we have to take into consideration, and that has to do with degradation being a slow process. So much so that the concentration degradation profile might look very different depending on incubation time. Here, we start out with the profile after 6 hours of incubating human PBMCs in the presence of different PROTAC concentrations. So PBMCs is just a type of cell type. And as you can see, now the hook effect fits these data nicely. So we do get results, right? But the question is, are they actually all that meaningful? After all, the choice to incubate for 6 hours is based more on convenience than any scientific reason. To address this issue, we use the protein's baseline half-life, so that is the turnover rate in the absence of drug, which is about 2 days in this case, together with that model we just fitted to make a prediction about what the concentration degradation profile would look like if we were to go with a longer incubation time of 24 hours. So we didn't measure degradation after 24 hours, but at least we haven't measured it yet. So we didn't use experimental data to get to the blue graph, but that's based solely on the 6-hour data, together with the protein's baseline half-life, which I looked up in the literature. Now, to confirm our prediction, we can now actually look at the real experimental data, which reveals that we have good agreement. Now, this goes to show that the hook model can not only be used to extrapolate the extent of protein degradation with regards to protein concentration, which is on the x-axis, but it also allows for extrapolation with regards to incubation time, so from black to blue. And most crucially, we can even predict what the steady state degradation parameters would look like, because it might be that the profile continues to change after 24 hours, right? I mean, who knows? Well, this table does. What it does is it tells you for how long you would need to incubate your PROTAC to get the real degradation parameters. That's the steady state values. of Dmax and DC50. And the time this takes depends on the protein's baseline half-life, so on the one hand side, and on the other hand side, it also depends on the PROTACs Dmax value. Now, we have a protein half-life of approximately 48 hours and a Dmax of at least 95%. So in our case, it would take at least roughly 36 hours, 36 hours of incubation, which is longer than the 24 hours we went for. The model, however, allows us to estimate the steady state parameters from a pre-steady state profile, like the one of the 6 hours of incubation. So we don't actually need to incubate for 36 hours. And that's a pretty good thing, right? Since it's questionable whether some of the longer incubation times, at least in this table, whether they even are experimentally accessible, just from a purely practical standpoint. So now we have successfully assessed this initial lead compound, so the black one. However, its degrader properties aren't all too impressive. So what we want to do next is we want to improve on them. So that is, we want to go from the black profile to something that looks more like the red one. And here, our KCAT model comes in handy, which predicts how the PROTACs binding affinities, how those need to be improved in order to achieve the desired degradation, right? So we want to go from black to red, that is, we want to go down and left. So down means more degradation, left means higher potency. Okay. I just said binding affinities that need to be improved, so that's the plural. Now, how many binding affinities are there? Well, of course, we have the PROTACs affinity for the target protein. Oh no, right. So now I think I just have to wait for the lag. So the PROTACs affinity for the target protein. And then we also have the PROTACs affinity for the E3 ligase, right? So that's the other binding partner. And then there's also a third affinity, and that is called a cooperativity, which quantifies the interactions between the target protein and the E3 ligase in the ternary complex, right? So we have a ternary complex, so 3 constituents, and that means there's 3 different interactions at play. Okay, so on the one hand, we have these 3 binding affinities, which are all compound specific. And on the other hand, we also have physiological parameters, which are specific for a given cell type, but more about them a bit later. Okay, now how do we get from the black profile, the bad one, to the red one, the good one? First, we need to determine the 3 binding affinities for compound A, so the bad one, to understand why it is so bad. And we can get those affinities either from biochemical assays or even from in silico predictions. In this case here, we use the experimentally determined parameters. But if you're interested in how our model can be combined with in silico AI models to predict degradation from chemical structures, there was an interesting poster at this year's PAGE conference where they did exactly that. And the link to that poster can be found in the bottom of this slide, right? So what they did is they used AI to predict the binding affinities from compound structure, and then they feed those binding affinities into our model. Anyhow, no matter whether we use measured or predicted affinities values, we then supply the KCAT model with that information before fitting it to the experimental concentration response profile, to the data we see here. And now, the model allows us to make an a priori prediction about how much the binding affinities would need to be improved to achieve the red profile. So the affinity for the target protein would need to be increased by a factor of 10 roughly, the affinity for the E3 ligase by a factor of 5, and the cooperativity factor by a factor of 1.5 to go from black to red. And as it turns out, medicinal chemists have been able to synthesize such a compound, so a compound with exactly those binding affinities, which was then run in this degradation assay. And as you can see, target degradation target degradation really was improved to the extent predicted, which validates our model. Now, that was all about coming up with predictions for different compounds, but always in the same cell line. In our case, that was Ramos cells. However, the model also allows to translate the results from this one cell line, which is shown in black in this slide, to other cell lines, so to THP1 cells or splenocytes, for example. And as you can see, we are able to correctly predict that there's more degradation in Ramos cells than in THP1 cells, or there's more degradation still in splenocytes, and that's always independent of the PROTAC used, right? So the same PROTAC has more degradation in splenocytes than in THP1 cells. And to understand why that is, we just have to turn to the expression levels of the E3 ligase and the target protein's half-life in these different cellular systems. So the more E3 ligase there is, the more degradation you get, and the longer the baseline protein half-life, and thus the slower protein resynthesis, the more degradation you get. Now, what this allows us to do is we can perform our experiments in a cell line, like the Ramos cells, for instance, which are easy to cultivate in vitro, so easy to do experiments on, and then we use the model to predict, based on the Ramos data, how much degradation we would expect in the primary cells in vivo, so in the splenocytes, which we might be actually more interested in. Now, moving on. So far, we've seen two very practical applications of the KCAT model, but we can also use it to generate some more, let's say, fundamental insights about the PROTAC mechanism of action, which is often described as event-driven, as opposed to occupancy-driven. Now, this plot here shows the relationship between degradation, which is on the y-axis, and this time it's really degradation, so it's not protein remaining, but degradation. Degradation after 24 hours of incubation on the y-axis, and target engagement on the x-axis, as it applies to all of these compounds in the two different cell lines that are shown. And so again, on the x-axis, we don't have PROTAC concentrations, but we actually have target engagement. And from this plot, we can learn several interesting lessons. For one, it takes only very little, so about 1% of target engagement to achieve 50% protein degradation. And what that means is that only 1% of the total target protein is part of a ternary complex at any point in time, and still you get 50% protein degradation. Now, this can help to understand why degradation takes so long, right? Because you need several rounds of engaging 1% at a time of the total target protein to degrade 50% in the end. Moreover, now if we inspect the graph closely, we find that initially, so to the very left, small changes in target engagement drastically increase the extent of degradation, while post a certain point, increasing target engagement any further, that will be of only very limited use. And during lead optimization now, it's crucial to determine whether your current front runner sits on the left or on the right in this plot, because that will determine how you should proceed with regards to compound optimization. Because remember, you can't just increase PROTAC concentrations to move to the right in the x-axis here, since the hook effect, right? Curve goes up again, introduces a limit to the maximal extent of target engagement. So for each compound, you have a certain maximal extent of target engagement. Now, if you're on the left of this plot, you can do, of the x-axis, you can do what was done here, and that is increase the binding affinities, including cooperativity of the compound. But if you're already on the right, you must turn to other optimization strategies that are less well understood, since there's no point in increasing target engagement any further because the graph won't go. And of course, the only way to know whether you are on the left or on the right in this plot is through modeling. Great. Now that's how we can predict what improvements to the binding affinities are necessary to achieve the desired degradation. But what actually is that desired degradation in the first place? Now, to answer this question, we have to turn to the third and final model of this presentation, which we call the PD model, of course. Now, what this model does is it translates a pre-specified effect on the level of the pharmacodynamic response, where we can easily come up with a target value for the drug effect, to an extent of degradation, right? So let's say we want to improve the initial lead compound in black to such an extent that its concentration PD response profile. So on the y-axis, we now have the downstream pharmacodynamic response rather than protein levels. So we want to improve the black profile so that it looks more like the green one, which has roughly 10 times greater potency. Right. And now what the PD model will do is it will tell us how the degradation properties, most notably DC50, need to be adjusted to get there. Yeah. Now, this table, the one to the left of the main plot in the slide, summarizes how well our initial lead compound, so compound X, the bad one, worked as a degrader. And the more of the target protein we degrade, the smaller the pharmacodynamic response. Here, that's about TNF-alpha release, right? So we want to inhibit the pharmacodynamic response. We want the graphs to go down. So the thing on the y-axis, again, that's not protein levels now. That's the downstream pharmacodynamic response. In this case, TNF-alpha response. And that should go down. And what we do now is we feed the information, so the DC50 and so on, the things in the table, into the PD model. And then we fit that to the experimental concentration PD response profile, so to the black data. To be decreased to get something like the green profile. And here that is by a factor of 3, roughly, if we also improve Dmax by a bit. And again, such a compound has been synthesized and the experimental data were in good agreement with our predictions. Now, the fact that all of this modeling worked out well, furthermore validates the target protein, as it really is degradation of that protein that explains a PD effect, right? So the same extent of degradation, no matter which of the two PROTACs you use, will always result in the same pharmacodynamic effect. Okay. Now, in the previous example, the PD effect was driven mainly by protein degradation, as the PD model shows. However, there are also cases where inhibition contributes to the overall effect, and there are some interesting phenomena associated with that. Now, you might be thinking, hold on, I thought we were talking about degraders here. Why is he talking about inhibition now? And you're right, of course. But see, the thing is that the moiety that binds to the target protein, for most PROTACs at least, that this moiety is based on a corresponding inhibitor. So the way this usually plays out is that there's an inhibitor project that aims at targeting a certain protein, but that project fails for whatever reason. And then a new project is initiated on the same target, but this time transforming the unsuccessful inhibitors into PROTACs. Like in this example here, which comes from an in-house project. Here, the solid line shows the concentration response profile for target protein degradation, which features the hook effect, which we talked about in great detail before. While the dashed graph is some sort of downstream pharmacodynamic response, and there we see no trace of the hook effect. As already alluded to, the reason for this observation is that at high concentrations, and you can see that in the small plot to the left, inhibition takes over from degradation as the main driver of the PD effect. So the contribution of inhibition to the overall effect increases with drug concentration. And that is completely in line with what the model predicts. So as long as the PROTAC is also an inhibitor, there should be no hook effect for the downstream response. Now, if you think about that, this might actually be an advantage of such PROTACs that are based on inhibitors, as opposed to PROTACs that are pure degraders that can't inhibit the protein they are targeting. Yeah. Now, finally, I want to show you another interesting observation, which we made in a different internal project, and that is about time courses. So what we did here is we first of all predicted the time course of protein degradation from the protein's half-life and its concentration degradation profile. And as you can tell from comparing our predictions of the blue graph to the observed data, so far everything has worked out pretty well. Next, we used the IC50 value, so the inhibitory potency of the E3 blocked control compound. So that's a compound that has all of the features of our protein, save that it cannot bind to the E3 ligase and thus cannot degrade the target protein. Now we used the inhibitory potency of that compound together with the known degradation time course to predict what the time course of the PD response would look like for our protein. And that's now so this prediction is the green graph. And again, we could validate this prediction. Now, when inspecting this plot, we note that the PD response is affected right away, while degradation is a relatively slow process. And therefore, and that's again shown in the plots to the small plots to the left, therefore, in the beginning, the PD effect is mainly driven by target inhibition, while target degradation only kicks in later on. Now, this might be another useful feature of PROTACs that are based on inhibitors, since they might be able to combine the rapid onset of action of conventional inhibitors with the extended pharmacology that comes with a protein degradation. So a lot of advantages for those repurposed inhibitors, as it seems. Now, to sum up, we have looked at, first of all, how to evaluate target protein degradation by PROTACs, where our hook model can capture the entire concentration response profile, and we saw how incubation time affects degradation and how to deal with that. Next, we discussed how we can use the KCAT model to predict what improvements in the 3 binding affinities are necessary to achieve a certain target degradation, and how our protein degradation in one cell type, how that translates to degradation in another one. And finally, we explored how the PD model can be used to translate target degradation to a more relevant downstream pharmacodynamic effect, where we know best what effect size we want. And we also touched on the benefits that target inhibition by the protein brings with it. Right? Goodie. That is, I'm afraid, all I have for today. But if you found this presentation interesting, or if you want to know how modeling can be used for the planning of animal PK/PD studies, so for going in vivo, then why don't you join us at the upcoming Protein Degradation and Targeting Undruggables Congress in Basel, Switzerland, which will be held at the 19th and 20th of September. So that's in 1 week's time. And I think there's more or less at the same time, there's also an M&S conference in Basel. Maybe you're already going to that. Anyhow, at this conference, the Protein Degradation Conference, my co-author and supervisor, Andreas Reichel, who's shown in the yellow box in the slide, will be giving a presentation, and he'll also be part of a discussion panel, while I will be there as well, with an accompanying poster, but without the beard, I'm afraid. Okay, so as I said, we presented analysis on both literature as well as in-house compound, and therefore I would be remiss if I didn't mention at least some of the great colleagues that generated these data and greatly inspired our modeling work. And of course, very special thanks also to go to my supervisor at ETH Zurich, that is Professor Kremer, for enabling me to conduct my PhD in this fashion. Now, with that, I am done with my presentation, and I am now looking forward to the discussion, and I think we should still have plenty of time. Thank you very much for your attention.
Thank you, Robin. And for our audience, you can ask questions to Robin by entering them into the questions pane on the GoToWebinar side panel. And let's start with the questions that we've already received first. And the first one is, how does the affinity for the ligase versus target protein impact the hook effect?
So as you can, you don't actually have to read the paper. If you could just follow the link, then I think we already get the view, so that will already be great. Anyhow, the way that works is, so the DC max, that's the nadir of this hook curve. That is located at the concentration, which is the geometric mean of the two binding affinities. So in other words, the smaller the affinity, so the greater the KD value, the further to the right that point is located. Yet it's also if the affinity is greater, so if the KD value is smaller, then the whole it looks a bit like a goblet, right? Then the whole goblet becomes wider. So, long story short, you want both binding affinities to be as small as possible. So, just like from the efficacy standpoint, in order to have the greatest potency and also not to worry, because then you don't have to worry about the hook effect so much.
Our next question is, what's the definition of TE% target engagement for a PROTAC? And since target turnover rate is critical for the degradation predictions, how is that value usually determined?
Okay.
So it's two questions.
Yeah. So I try to, but maybe you'll have to remind me of the second one as I addressed the first one. So target engagement, exactly, that's the TE thing. And the way we define that, and actually that's also kind of interesting, because coming from inhibitors, right? For inhibitors, you usually have one drug and one target. And then it's quite easy to say whether the target is engaged or not, because if the drug is bound to the target, then the target is engaged, right? But with PROTACs, you have two targets. And the way we define target engagement is if both of these targets are engaged. So TE, in other words, is the fraction of the total target protein that is bound in a ternary complex. And the second question, what was the second question again?
Target turnover rate, and how is that usually determined?
Okay, yeah, so there's the way we do it, at least, is that there's two methods that we commonly employ. One of them, actually three. So one of them is just looking it up in literature. But if we want to measure it ourselves, then there's two methods. One of them is to determine the degradation time course. So what you do is you take your cell culture, and then you add a certain concentration of PROTAC, and then you track over time how protein levels decrease. And from that profile, which is a exponential decay towards a limit value, from that profile, you can calculate the baseline protein turnover. So not only the turnover that's induced by the project, but also the baseline turnover. That's the one approach. The other one would be stable isotope labeling of amino acids and related approaches. So for example, there's also the cyclohexamide chase assay, but what all of those have in common is that you somehow label the target protein, and then you look at how the labeled fraction of your target protein decreases over time, right? And that tells you how much of the target protein is still the labeled one and how much has been resynthesized since you labeled the protein.
So regarding the PD model, why do the simulation results have different initial values? Shouldn't they be the same at the beginning because there's no PROTAC yet?
So yes, you would expect that, wouldn't you? The thing is that, so we used the real experimental data there, and what we saw is that at very low PROTAC concentrations, the protein levels were higher than in the control group, right? I mean, that is most likely because the control group, so that just was random variation in the control group. So the control group would go. So that is most likely the explanation, right? So because if you repeat the experiment without adding any PROTAC, then you will still get, since these assays are quite variable, you will still get different values each time. And if the value that you normalize your data to is just by chance a bit lower than all of the other values of low concentration, then your effects at very low concentrations will be greater than 100%.
Are there any pesticides on the market that work through PROTAC degradation?
I am terribly sorry, I have no idea.
Okay. How much hook effect do you think will appear in vivo, as if high concentration inhibition is driving the process, you may not have a hook effect at all?
Yes, so I mean, that is of course absolutely, so I think there's nothing I could add to that other than there is an IQ consortium paper that came out. I don't know whether that's come out already, but that's at least like in preprint status. And there are different pharma companies that they encountered the hook effect in vivo, and a small fraction of those that they did, and that interfered with their analysis. So what I would say is that you really have nothing to lose if you just consider it. You know, if you use this hook model thingy, because then you are aware of what might happen at high concentrations, and you don't have to worry about whether your model is actually wrong. But of course, as you said, if the concentrations stay well below the DC max, then there's no hook effect in vivo then.
Has your models been applied to describe clinical PK/PD work?
No clinical data. So we use it at Bayer. We don't have any projects in clinical trials as of yet, and we only use it on animal studies.
Is it best to have both KDs of the bicomplex within the same target range?
So I would say no, but that now goes into more strategic than really modeling discussions, because there's a benefit to having a relatively high affinity for the target protein and a somewhat lower affinity for the E3 ligase, and that is if your affinity for the target protein is higher than that for the E3 ligase, then you should, in theory, have less off-target degradation, right? Because if already the affinity for the E3 ligase is very high, then also a lower affinity for potential off-targets would result in degradation of those off-targets. And so you get an additional safety margin if you have high affinity for the target protein, relatively low affinity for the E3 ligase. But then again, I wouldn't necessarily recommend to ever lower any of the two binding affinities on purpose, because it's kind of hard to get good affinities anyway.
So the turnover of a protein impacts the efficiency of the potential PROTAC. Did you perform a sensitivity analysis to determine a critical turnover time at which it would make sense to go for a PROTAC approach?
Yeah, so the thing is, you need to know, so that one of the models was called the K-cat model, and that is because of this parameter K-cat, which is the rate of presumably target protein ubiquitination. And the critical parameter is, or the critical metric, is the ratio of this rate of target protein ubiquitination, so the ratio of that and the baseline protein turnover. Right. And in our case, I think, well, let me think, but I think, so this TE50 value, which was 1%, that is somehow related to that ratio. So I think that the protein ubiquitination rate is 100 times faster than the baseline protein turnover rate. But you can read that up in the paper if you're interested. And with that, so if this ratio is that great, then you only need 1% of target engagement. And now you can think of, if it would be even greater, then you would need less of target engagement, whereas if it's lower, then you need more target engagement.
So, and this is actually a follow-on, with the diminishing return for target engagement, would you possibly want lower binding affinities in some PROTACs to reduce the target engagement?
I don't think so. So maybe whoever wrote that question, maybe you could. So why would you? So I think you should never tell the chemist to make a compound that is worse, kind of with regards to protein degradation, because again, that's pretty difficult to get a good protein degradation, and so I mean, you could think of scenarios where it would make sense from a modeling point of view, but just keeping in mind, like everyday project work, I think if you tell the chemists to make the drug have lower affinity for the targets, then I would be quite surprised.
So this next question is sort of a combination of questions. I'll read the whole thing first. Is alpha estimation identifiable? Are you using SPR binding affinities and estimating alpha, or do you have any experimental measurement of alpha?
Mm-hmm. So if you're talking about identifiability, then I should maybe reiterate that what we did here is not that we informed these binding affinities from the degradation data, right? But we measured binding affinities in orthogonal assays, and then we used those affinities, which we already knew beforehand, to predict what degradation will look like, right? So we're not going from data to model, but rather from model to data. And the way these parameters were measured, and those measurements were performed by colleagues called Sorba at PAL. And the way they performed their measurements was using a surface plasmon resonance for the binary binding affinities. And then I think TR FRET, which is, I think, time resolved first resonance energy transfer or something, to also determine the cooperativity value. So those are all biophysical assays that were performed. And you can like, if you're interested in these methods, then there's a great they are outlined in great detail in this publication that we also cited throughout the presentation. Was that already all of the questions, or did I miss one?
I think so. There is another question. I'm going to ask it. I'll read it as it's written. How did the chemistry team dial in the parameters? Was there a series of in silico models that helped with this, or was it intuition and trial and error?
So I think it was by increasing the length of the linker. So they used, but again, that's if you're interested in more detail, then there's a publication on that. And also, but the way they achieved their goal was by so they have a PEC linker, so linker, and they just added one after another polymer residues to like increase the linker length by one increment at a time. And then you get better affinity for both binary affinities improve, but also cooperativity improves. And you can think of it like if the E3 ligase binding moiety and the target protein binding moiety are further apart, then there's less steric clashes between the two proteins.
For our audience members who asked about follow-up contact, we will send your information to Robin so that you can contact him, or he can contact you on the follow-up. We have one final question, which relates to, you mentioned using failed inhibitors in the PROTAC. If they failed as and this is, sorry, it's a two-part question. If they failed as an inhibitor, would that mean that they just didn't have the affinities necessary, and therefore you shouldn't take the inhibition into account when you look at their abilities, or they just weren't a good enough inhibitor?
So the latter. The inhibitors. Well, I mean, I can at least talk for so there was one example from an internal project which I showed, and there I know like more of the background story as well. And there, so they worked, inhibitors basically worked as inhibitors, but they had unfavorable properties. So for example, they might have too low binding affinity, but it might also be that their PK. So for inhibitors, inhibitors are occupancy driven, right? So what that means is you need exposure all the time to have an effect. So as soon as your drug concentration, your unbound drug concentration in plasma drops below the required efficacy threshold, you will basically lose you will lose your effect. Whereas for PROTACs, you have an extension of effect due to this is called extended pharmacology. So due to the fact that once degraded, the protein has to slowly be resynthesized before any downstream effects can return. And so that is like that is a very PK/PD modeling interpretation-centric reason for why you should convert a bad inhibitor into a PROTAC. Another reason might be that it's just a new patent matter, you know. So if you change the molecule from an inhibitor and then make it into a PROTAC, then you can patent that again. But that's more of a boring reason, I would say.
Thank you. And thank you, Robin, for a very interesting presentation and a very interesting discussion as well on this topic. And again, we'll pass on the information from the audience to contact them. With that, I'd like to thank you for a very interesting webinar, and we look forward to perhaps having you back again as your work continues. Thanks to our audience, and we hope to see everybody again at a future webinar. Thanks, Robin.
Thank you, Rebecca. Bye.